Thank you, Peter, and thank you for inviting me here. I was also very pleased to be introduced as a young scientist, that was quite a few years ago. I will talk about cartilage and stem cells, and giving the perspective of regenerative medicine. But first, the musculoskeletal disease which you all represent are working with has a huge impact on society. These are just a few figures here. In the US, the total cost for musculoskeletal disease is about $850 billion US. And there's a high prevalence, and with increasing ages of the population, we will have even higher numbers of this. The interesting thing is also that the research budget for the NIH for this field, which actually affects a lot of people, is just 2% of the total budget, which is something we should acknowledge. I would like to put this into a perspective. When we talk about regenerative medicine and tissue engineering, and this is what we're talking in osteoarthritis, but this is also valid for Alzheimer's disease or other chronic diseases, is that there is a very long time period for the initiation of disease and the end stage. And what we have been working with so far for cell therapies and others is the tissue engineering part when we have defects, when we have osteoarthritis, where we have to replace the joint. And I'm an MD, and for a doctor to do a treatment, you have to know what the disease is. You have to make a diagnosis. And the problem is that when the disease is there, it's obvious, and the diagnosis is easy. But to do the regenerative medicine part, which we all are walking into, with early treatments, you have to know that there actually is a disease. And today, for all of these chronic diseases, there is no early diagnosis. And that means also that there is difficult to distinguish a patient at an early stage who should have the treatment and who should have not. And I will come back to that. If you look at cartilage repair, the capacity of regeneration of an injury diminishes with age. If you have a fetal cartilage, you see here, this is a sheep, you have a good regeneration capacity. On the other hand, the oral cartilage has very limited capacity of regeneration. The question is why? We have inability of filling out the defect because there's no blood system that will provide a clot. There are very few repair component cells, and most of them are dying for other reasons. And there are really no chemotactic signals. And the cells that could repair, they are trapped in their matrix. The current treatments of conventional treatment is that you do drilling or microfracturing, where you recruit cells from the bone marrow. There have been combinations of other tissues, as well as osteochondral graft, where you take a piece of cartilage and put that into the defect, or autologous chondrocyte transplantation, which was developed by our group together with Lars Peterson and Mats Brittberg in the late 1980s. These are chondrocytes, and the question is, are they capable of regenerating cartilage? And what you see here are quite intelligent guys. What you see now is a 5-hour time frame where you see the cells. They talk to each other, they move, they have quite interesting conversations. You see here in the corner, there's one guy coming here, obviously attracted by these other cells. And this is very random, and of course, like Sam said, if you could organize this with nanofibers, they will probably do a much better coordinated work. But these are the cells that you implant into the defect. So the question is this working? We introduced this, we had animal models showing regenerative capacity in rabbits, similar to what Sam demonstrated with his nanofibers. And the basic concept is actually if you have a defect, you take a cartilage biopsy, you isolate the cells, you culture the cells together with the patient's serum, and then you implant the cell under periosteal flap, which was the original design. We have published several papers over the years, and recently we were able to take the 15 to 20 year follow-up of our patient group. This was in 2000, where we demonstrated that if you have a good clinical outcome after 2 years, the clinical outcome will last at least 10 to 15 years later, 5 to 10 years later, sorry. And this is 10 to 20 years follow-up of 250 patients gave a good clinical result, and 91% of the patients wanted to have the operation done again, if necessary. We also have taken biopsies earlier, but it's not ethical today. This is a paper where we compared with gadolinium enhanced MRI, where the cartilage defect in 10 to 20 years after the transplantation has similar characteristics as the surrounding cartilage, although there are patients that have had macrofracturing have problems with subchondral bone. But Mats Pripyat has already talked a lot of this yesterday, I think. So I will go into the future concepts. So today we have treated over 1,700 patients here in Sweden. There is a long time clinical durability. Worldwide, it's about 30,000 patients treated. It's approved by FDA. Today, the original method is not used in that sense. It's combined with matrix or MACI or hyalurograft C. I will come back to that. And there are randomized clinical trials. 3 out of 4 studies have demonstrated superiority over conventional treatments, although it's still debated. But there's no off-the-shelf product, which means that it's not available to many patients. And what we'll see in Europe here is that we have a regulatory environment which will further complicate the future development of cell therapies. The second generation of culture cartilages, and which is basically used now by my colleagues, is that you culture the chondrocytes on a hyaluronic acid scaffold, which will enable an implant by arthroscopic procedure, which gives a much better clinical outcome for the patient. And Mats Pripyat is currently conducting a follow-up of the first 100 patients that has passed 2 years. The third generation of cell therapies would be a universal donor cell product, which is produced and you can have it off the shelf, take it and implant into the patient. And that will be a combination of scaffolds, nanofibers, hyaluronic acid-based or collagen gels, and some other cell type, which is not determined yet. It could be mesenchymal stem cells, orbital stem cells. It could be progenitor cells from human embryonic stem cells or umbilical cord or cord blood, and I will address a few of those here. But there are scientific challenges. First of all, the mesenchymal stem cells, which is easily available, has a default pathway of bone differentiation. It doesn't stop at hyaline cartilage, it continues with hypertrophic cartilage. And the same goes for umbilical cord, but it's not totally determined. It's possible to derive chondrocytes from human embryonic stem cells, which I will show you. But the question is, do we have a stem cell-free implant so we don't get a teratoma? And that's still not proven convincingly. And then orbital stem cells, do we have a stem cell population within the cartilage that can be expanded at a large scale? So do we have stem cells in cartilage? And I will try to address that. And last, it has been determined that the joint is a separate lineage of cells rising from a GDF5 expressing cell population that produced the joint and the surrounding structure. So it's actually a unique inherited population. And the question is, can we take cells from other places to produce cartilage? Mesenchymal stem cells has multi-potential. You can produce chondrocyte, bone, as well as fat cells. But if you do take articular chondrocytes, it has the same capacity, but the default pathway is cartilage and not bone, as in the case of mesenchymal stem cell. This was work that was done with one of my PhD students together with Arnold Kaplan. 10 years ago, the embryonic stem cell came into the focus of everybody's mind. And I have been working with and had the idea perhaps these can be used to produce chondroprogenitor cells. It's possible to derive neural cells. You can derive any type, theoretically, of the body's 250 different types from an embryonic stem cell. This is a paper we published last year where we derived embryonic chondroprogenitor cells from embryonic stem cells by means of co-culture. It's not possible to derive the cells by just taking the culture medium or condition media from chondrocytes. You have to have physical contact. So what we did was that this is the pellet mass culture, a high-density culture of chondrocytes, and you have irradiated chondrocytes. These cells can be propagated further if you release the cells on monolayer culture. Irradiated cells, they will die. Embryonic stem cells, if put in a pellet high-density culture, form a cystic structure, and it dies at second passage. But if you combine the two cells, irradiated chondrocytes with embryonic stem cell, it forms a chondrogenic structure. The cells survive and can be propagated and are actually able to form chondrocytes in pellet mass. This is the embryonic stem cell originally. This is the co-cultured cell type. And it can survive and produce cartilage on a matrix, and it also produces colonies of cells in agarose culture. So it's possible to derive chondrocytes, but it's still not at the highest level of normal chondrocytes that we take from the patient. So then we have the question. So these are the cell therapy aspects. And the second part is, how do we address this frame here? How do we find the disease at a stage where we can intervene? So can we increase the diagnostic potential, and can we understand actually what the disease is? There are no modifying treatments, disease-modifying treatment in osteoarthritis, except perhaps that cell therapy could regenerate cartilage. So the question is why? The reason is that the disease is not diagnosed until the progression and pronounced alterations in the joint lead to pain and radiographically detected changes. And there's a biomarker missing, which means that the pharma industry is totally uninterested in producing a clinical trial for any type of substance and wait for an X-ray 25 years later. But if we had a biomarker, the story would be different. So in regenerative medicine, we think about the tissue as a tissue equilibrium, where you have recruitment of cells that supports the organ over time, and we have cell death. And this is valid for a lot of organs. You have the skin, for instance. Half of the dust you have at home is actually your family's skin that is discarded. It's renewed every third week. You have an intestine that produces about 0.5 ton of intestine during your lifetime. So we have high capacity, and recently the brain and the heart has also been demonstrated to have this. The question is, how is this regeneration maintained? You have to understand stem cell biology, and the driving force between regeneration is actually a stem cell niche. It's a microanatomic structure where the stem cells are situated under control of surrounding usually mesenchymal cells, and they are released and producing and supporting the tissue or choice. There are a few well-defined stem cell niches. One is in the intestine, and another is in the bulge stem cell area of the hair papilla. But the interesting thing, if you look into these structure, which has been scientifically very well characterized, is that they have about the same signaling system that supports the growth. And the interesting thing is that they are highly conserved systems, which can be found in the fruit fly, it can be found in very primitive insects as well as animals, and I will come back to that. So the question is, do we have stem cells in cartilage? We isolated, this was the phenotypic plasticity which I demonstrated earlier with the mesenchymal stem cell. So chondrocyte has pluripotency. This is also valid if you take a single cell of chondrocyte and expand that, it will end up with chondrogenic tissue, adipogenic, or osteogenic tissue. As this is just to illustrate that I'm not that young. This was a paper we presented in 1992 illustrating the stem cells and the effect of growth hormone. And for you who believe that there was an era before PowerPoint, this is what to demonstrate this. This was actually a drawing which we at that time had. But the interesting thing about that, and this actually came back, because then there was no stem cell niche in discussion. But we demonstrated using tritiated thymidine that we have label-retaining cells in the growth plate of the rat, as well as on the surface of cartilage and also in this region, which is called the zone of Ranvier. So we repeated this experiment now with another pair of glasses and subjected a rabbit with BRDU, which is incorporated into the DNA, and were able to find a stem cell niche in the area of close to the joint, but close to the growth plate. And we also found label-retaining cells in the articular cartilage interspread and in the growth plate where they were supposed to be found. This niche also expressed a lot of stem cell niche markers, which is found in the niche of the hair as well as the intestine. I'll not go into this. And the same goes for articular cartilage, which has, for instance, notch expressed on the surface layer. And interestingly enough, the cells here are lined with GDF5 expressing cell, which is the lineage of the articular cartilage. So this gives indication of, this is a hypothetical structure that we have found. It supports definitely the stem cells of the growth plate, but there's also a suggested migration into the articular cartilage, and this is still an effort we are working with. We have also, together with Helena Brisby as a collaborative work, found a similar structure in the spine and supporting the annulus fibrosus and the nucleus. Finally, we were looking into these. Why are a tissue of cartilage having a lot of conserved regulatory signaling patterns from stem cells? There's one hypothesis produced by Charlie Archer, where the notch cells, which are expressed on the surface, adds new cartilage. This is a stem cell that will add new cartilage by appositional growth. This is one hypothesis. And the new cells are coming from the surface. The notch system is actually a discovery from 1917, over 100 years ago, when you looked at fruit fly. And a few of the fruit flies had a little notch in their wing. And this gene was sequenced 1980. It was about 60 years later. And now it's definitely a very interesting signaling pathway that controls cell-to-cell action, but also controls stem cell differentiation. So we looked into this. Perhaps if it's there in the normal cartilage, where we see the notch receptor, we have its ligand, and we have also HES5, which is the downstream gene. So it's in the surface of the cartilage. But the interesting thing is, if you look into osteoarthritic cartilage, you have a very high up of the signaling system as a part of the disease mechanism. So that's an interesting finding. We also have now a unique capacity to actually look at the whole genome expression by microarray data. So we did a genome-wide scan comparing osteoarthritic cells with normal cartilage. This was done in collaborative work with Charité in Berlin. And these are just illustrating the normal cartilage from deceased donors, osteoarthritic cartilage. This just gives a clustering data saying that these two populations are distinct from each other. You get about a couple of 100,000 data points. You cannot present an Excel sheet. You have to use something else. This is an intelligent program where you actually look at the signals and you combine data which is generated from literature. And you can see what's upregulated. You can also see if you have a lot of interaction between different signaling pathways. You can say what's downregulated. You find specific hub genes. But basically, if you combine this, you see there are lots of angiogenesis in osteoarthritis, there are lots of apoptosis, a lot of chemokines, a lot of inflammation, etc. But we also looked at the signal transduction, specifically the Wnt signaling pathway, which is the other control system for the stem cells. And interestingly, I will not dwell on that. This is work produced by one of our PhD students, Anna Torfe, together with Camilla Karlsson. But basically, this system is also activated in osteoarthritis. Red means up-regulated and green means down-regulated. So what we have here is a disease, is osteoarthritis, where we have a lot of activation in very primitive stem cell regulatory pathways. So that's puzzling. But even more puzzling is that when you look at genome links to osteoarthritis, you would expect that there are matrix components. For instance, if you have type 2 collagen mutations, you will get osteoarthritis when you are 15 years old. But that's single gene disease. It's a monogenetic disorder. But if you take a population, what do you find? There are a few very convincing ones. One is GDF5 is connected to osteoarthritis. The other thing is that iodothyronine deodorinase enzyme type 2, this has to do with the conversion of T4 to T3, which is the thyroid system. But it's also involved in stem cell control, but it's also, for instance, it's turned on and turned off during retinal development. So here again, we have a developmental gene connected to osteoarthritis. And HIP-OA in women are associated with frisson-related protein, which is the Wnt system. And finally, a very recent study with about 6,000 individuals, very well done, also found another linkage, G-coupled protein receptor 22, GPR2, function unknown, but it's co-localized with notch in the surface of articular cartilage, but is not found in cartilage. So what we see here is that the genetic linkage is to stem cell control, which will give things for the future. So, conclusions: chondrocyte transplantation is some type of a disease-modifying treatment 10 to 15 years later. It's possible to derive universal donor cell lines, but no one has been convincingly developed, although embryonic stem cell has the potential, but there are probably others. Stem cell signaling in the Notch and Wnt system is involved in osteoarthritis, and there's also a genetic linkage to stem cell homeostasis and control in population studies. So there is a potential of developing new disease-modifying drugs. And there is a stem cell niche that we found in the rabbit joint and in the intervertebral disc region, but we need to discover its function and migration of cells from that remains to be elucidated. And early osteoarthritic markers has to be determined, and I think that we have collaborative work with Rick Hanegård and Lund that there is a possibility that by looking at specific cleavage sites based on the inflammation in osteoarthritis, you can discover peptides which are not present in the normal cartilage, and thereby perhaps having an early diagnosis, and by that also giving a potential future treatment. And I would just like to acknowledge my collaborators for many years, Lars Petersen and Mats Brittberg. A lot of the data for ESLs is from Armin. Camilla did a lot of the genome gene array, and we have funding from Swedish Research Council, Gothenburg University. We had a 6th framework program, and some other funding. Okay, thank you very much.
Thank you very much.